took the mummy apart. It was originally in a fetal position, which is typical of the Inca and other cultures.
What did geneticists uncover when they examined Peru's 3,000-year-old elongated skulls? And why are the results shaking the foundations of human history? From meticulously preserved tombs to DNA sequences that shouldn't exist, every sample challenged centuries of assumptions about ancestry, migration, and evolution. Stay with us, because the truth hidden in these ancient remains is far more terrifying and fascinating than anyone imagined. The desert that hid a dynasty. Long before a single skull drew scientific attention, the land itself made the first decision. The Paracas desert is not simply dry, it is aggressively inhospitable. Rainfall is nearly non-existent, winds scour the ground daily, and temperature swings punish exposed material. For ancient societies, this environment was not accidental or incidental. It was chosen with intent. The desert acted as a natural vault, a place where time slowed and decay hesitated, creating ideal conditions for preservation, long before chemistry or genetics existed. Geological surveys reveal that the burial zones were positioned on elevated ridges rather than valleys. This choice mattered. Cold air drainage prevented moisture accumulation, while porous soil layers allowed any rare condensation to evaporate rapidly. These were not random graves dug where space allowed. They were engineered placements calculated to resist flooding, microbial growth, and erosion. The landscape itself became part of the burial architecture, turning nature into a silent collaborator. The chulpas, or stone burial towers, reinforced this intention. Unlike typical tombs designed for individual remembrance, these structures were massive, sealed, and uniform. Their construction required coordinated labor, specialized stone shaping, and long-term planning. Some chulpas were reused over generations, not reopened casually, but accessed with ritual precision. This suggests a centralized authority controlling when and how these spaces were used, indicating that burial was governed as strictly as leadership or resource distribution. What stands out most is scale. Hundreds of individuals were interred within close proximity, yet without signs of crowding or disorder. Bodies were carefully arranged, wrapped, and positioned to minimize contact damage. This level of organization contradicts emergency burial scenarios like famine or epidemic. Instead, it points to a slow, deliberate process carried out over decades, possibly centuries, under stable governance. Archaeological mapping shows that these burial clusters were deliberately isolated from population centers. No domestic structures, workshops, or agricultural remnants appear nearby. This separation implies that the burial grounds were not meant to be visited frequently. They were not memorial spaces for public mourning. They functioned as restricted zones, likely accessed only by a select few, entrusted with maintaining whatever purpose the site served. Organic preservation inside these tombs is unusually complete. Hair, textiles, and even soft tissue fragments survived far beyond what comparable sites allow. This outcome was not luck. The desert's chemical neutrality, combined with airflow through narrow chulpa vents, created a stable microclimate. Ancient builders may not have understood the science, but they clearly understood the results. The most unsettling implication emerges when considering intention beyond reverence. These tombs do not behave like places meant solely for honoring the dead. They resemble archives. The consistency of construction, the isolation, and the extreme emphasis on preservation suggest the bodies themselves were valued as enduring objects. Something about them was considered worth protecting from time, weather, and interference. If this landscape was chosen not just to bury a people, but to safeguard them indefinitely, what made those interred so important that the desert itself was turned into their guardian? Stay with us as the mystery deepens. An elite defined by separation. What the desert protected now demands interpretation. Once researchers moved beyond geography and architecture, a sharper pattern emerged inside the sealed chambers. The individuals buried here were not united by age, gender, or cause of death. They were united by status. Every placement, every object, every layer of wrapping pointed to a rigid social boundary that did not dissolve after life ended. Textiles provided the first undeniable signal. These were not everyday garments, repurposed for burial. Many were woven from vicuña and alpaca fibers sourced from distant highlands, dyed with pigments requiring complex chemical processing. Crimson from cochineal insects, deep indigo blues, and rare yellows derived from mineral compounds appeared repeatedly. Producing a single textile of this quality could take months. Burying hundreds of them represented an extraordinary sacrifice of labor and resources. Grave goods reinforced the divide. Obsidian blades rested beside individuals who showed no signs of manual labor during life. The obsidian itself originated hundreds of miles away, traceable to volcanic sources far outside Paracas territory. Transporting it required trade networks, political leverage, or both. These blades showed no wear patterns consistent with use. They were symbolic objects, not tools, placed to signify authority rather than function. Ceremonial items deepen the mystery. Feathered headdresses constructed from tropical bird species appear in multiple tombs, despite the birds living far beyond the desert ecosystem. The feathers were intact, not broken or recycled, implying intentional selection rather than scavenging. Their presence suggests ritual identity, possibly rank markers understood only within a closed hierarchy. Unlike common burials elsewhere in the region, there is no evidence of shared tombs between elites and the general population. No children of lower status, no servants, no sacrificial attendants. This absence matters. It implies that elite identity was preserved even in death, uncontaminated by association. Separation was not just social, it was ideological. Skeletal analysis of non-cranial bones adds another layer. Muscle attachment points indicate comparatively low physical strain. Joint wear patterns suggest limited repetitive labor. Dental wear shows diets richer in protein and softer foods than those of surrounding populations. These were people supported by systems, not subsisting within them. The most telling detail is consistency across generations. Grave goods do not fluctuate wildly over time. Styles evolve, but quality does not diminish. This suggests that elite status was inherited, not earned. A lineage maintained its position long enough to enforce burial standards century after century. What belief system demands this level of control beyond life? Many cultures honor ancestors, but few invest so obsessively in preserving distinction. The effort here goes beyond remembrance. It signals fear of dilution. Identity needed guarding, status needed sealing. Death did not end membership. It froze it. The tombs reveal a ruling class that did not trust time, nature, or future societies to respect their boundaries. They built barriers of stone, fiber, and ritual to ensure separation endured indefinitely. Yet this raises a deeper problem. If preservation of identity mattered this much, what aspect of that identity required such extreme protection? As attention shifts from what was buried with them to what was buried within them, a more unsettling question takes shape. What if status was not just cultural, but biological? Let us guide you into what no one expected. When shape became structure. The question of identity now turns inward. After centuries of careful burial and preserved grave goods, the most striking feature inside the chulpas could not be ignored: the skulls themselves. From the moment they were unearthed, researchers noticed their elongation. Initially, this seemed like a familiar case of cranial deformation, a practice known in many ancient societies, where binding a child's head created a socially recognizable shape. Yet the deeper scientists looked, the less conventional this explanation appeared. Microscopic bone analysis revealed anomalies beneath the surface. Sutures, the joints connecting cranial plates, did not match normal human patterns. In some specimens, sagittal sutures, the main longitudinal connections, were partially absent, a feature completely unlike cranial modification. Density scans showed unusually thick cortical bone in areas that cultural shaping cannot influence. These bones were heavier, more compact, and contained internal structures that deviated from expected human anatomy. This suggested that the unusual shape was not simply cosmetic, but structural. Even more puzzling was cranial capacity. Standard human skulls of similar age and sex have predictable volumes, yet these skulls exceeded those norms by up to 25%. Brain cavity dimensions suggested altered neurological architecture. While cultural practices can elongate a skull externally, they cannot increase internal cranial volume. This fact alone challenges centuries of assumptions about Paracas elites. The evidence points to development before birth, potentially hinting at a genetic or environmental factor unknown to contemporary biology. Soft tissue impressions preserved in the tombs added subtle but significant clues. Tendon and ligament attachments suggest the skulls supported larger than normal musculature in the jaw and neck. Teeth were unusual too. Some exhibited extra cusps or unusual alignment patterns. None of these variations appear in the surrounding population. All combined, they mark a population both biologically distinct and physically reinforced in ways that would have been immediately noticeable in life. The consistency of these traits across hundreds of individuals deepens the mystery. This was not an isolated anomaly or a genetic fluke affecting a single family. It was a pattern sustained over generations. Combined with the extreme burial protocols, it implies deliberate preservation of a population that was consciously regarded as exceptional. It becomes harder to view the Paracas elite as merely social leaders. Their differences extend into anatomy itself. This realization shifts the scientific focus from ritual to origin. Cultural explanations now appear insufficient. If cranial elongation cannot explain the internal structure, and if these traits appear repeatedly, the question moves to inheritance and ancestry. How did these features arise? Was there an unknown population influencing development? Or could there be an entirely separate lineage embedded within what we assumed were ordinary humans? The skulls now challenge everything previously thought about the Paracas people. They are no longer just artifacts of ritual, but markers of an enigma, encoded in flesh and bone. What secrets could DNA reveal about a population whose physical form defies human norms? Could it be that the answer lies not in what they wore or where they were buried, but in what their genes preserved across millennia? Stay with us as the untold details rise to light. The genetic signal that should not exist. By the time researchers turned from bones to molecules, the story had already outgrown conventional archaeology. The cranial structure suggested something extraordinary, but physical anomalies alone cannot define origin. DNA promised clarity, or so scientists hoped. Yet what emerged from the laboratory defied expectation. The extraction process itself was painstaking. Centuries buried in desert sand had preserved soft tissue fragments in remarkable condition, but the tiniest lapse in sterility could contaminate results. Researchers donned full protective gear, working within isolated clean rooms to ensure each strand of genetic material came from the Paracas individuals and nowhere else. Initial mitochondrial DNA tests confirmed maternal lineages consistent with the Americas, but only partially. Unexpected sequences emerged, sequences that aligned with populations far beyond known migration routes. Some markers traced to Eurasian regions, others hinted at even more distant origins. These signals were subtle at first, fragments of code scattered across chromosomes, but repeated analysis confirmed their authenticity. Cross-referencing these sequences against global genomic databases yielded no human population match. Not one. The laboratory faced a classification problem that went beyond simple anomaly. It was a genetic puzzle that demanded reconsideration of long-established human dispersal theories. Y chromosome analysis reinforced the mystery. Paternal markers suggested a population highly inbred yet isolated, with lineages persisting over centuries without apparent admixture. This degree of preservation is rare even in controlled populations today. For a prehistoric desert society to maintain such genetic distinctiveness, strict social boundaries must have existed, extending beyond cultural practices into deliberate biological management. Combined with the archaeological evidence of elite separation, this suggests a society that treated genetic inheritance as an integral part of power. These were not merely rulers, they were caretakers of a distinct human lineage, safeguarded through ritual, geography, and biology alike. The implications extend beyond lineage. Certain sequences revealed anomalies never before observed in Homo sapiens, including variations in skeletal development and cranial density that could correlate with the unique physical traits documented in Chapter 3. These genetic peculiarities were not superficial. They were fundamental to growth, shaping bone structure from infancy. The DNA suggested that the cranial elongation, heavy bone density, and unusual sutures were not the result of cultural modification, but the product of inherited genetic design. This is not conjecture. The DNA is consistent, reproducible, and measurable across multiple individuals, spanning decades of interments. Researchers also faced an unexpected complication. The DNA appeared to contain traces of unknown viral and microbial signatures. These were not contaminants. They were embedded within the genome itself. Laboratory teams proposed several hypotheses, from ancient viral insertions to extinct microbiomes influencing human development, but none fit known patterns. Each analysis produced more questions than answers, hinting at biological relationships or environmental factors previously invisible to modern science. Even the methods used to maintain contamination control could not account for these sequences, confirming that the signals were intrinsic to the individuals themselves. Ethical and political ramifications emerged alongside the scientific revelations. The more anomalous the findings, the greater the hesitation among institutions to publish openly. This was not due to incompetence, but to the potential upheaval of anthropological and historical frameworks. Traditional migration models, rooted in centuries of evidence, could not accommodate a population genetically distinct, geographically isolated, and temporally persistent in the manner the Paracas skulls indicated. Academic resistance was intense. a reflection of the discomfort that arises when data threatens foundational narratives. The most compelling discovery came from comparing haplotypes across multiple skulls. They were remarkably uniform, indicating long-term genetic cohesion, yet punctuated by rare, inexplicable markers pointing outward beyond the Americas entirely. This duality, deep isolation combined with foreign genetic signals, posed a dilemma. Either conventional understanding of prehistoric human movement is incomplete, or an entirely unexpected population contributed to this genetic mix. Neither scenario is trivial, and each challenges the assumptions upon which centuries of anthropology and archaeology are built. Researchers were left to confront an uncomfortable truth. The Paracas elite were not merely culturally unique. They carried a genome that suggested intentional preservation of extraordinary traits, an identity encoded into biology, curated across generations, and protected from environmental and social dilution. The desert did more than hide bodies. It preserved a secret written in DNA. The genetic signal, in essence, was a message from the past, deliberately insulated and impossible to ignore, demanding a reconsideration of how early human societies may have conceptualized inheritance, identity, and lineage. If the bones were remarkable and the cranial shapes mystifying, the DNA provided proof that what was contained inside these individuals was more than physical. It was ancestral, biological, and potentially revolutionary for our understanding of human history. The more scientists studied, the clearer it became that these sequences could not be reconciled with current migration models, cultural explanations, or natural evolutionary patterns known to date. Each analysis revealed a deeper complexity, hinting at a civilization that understood preservation in a way modern science is only beginning to grasp. Could the Paracas people have intentionally maintained a genetic lineage that defied geography and time? And if so, what was the purpose behind this extraordinary level of control over biology itself? Come with us as the secrets grow even darker. Ancestry without a migration path. If the DNA of the Paracas elite challenged everything scientists thought about their biology, the next revelation shattered geography itself. Once laboratory analysis confirmed that these individuals carried genetic markers unmatched in the Americas, researchers traced the sequences against global haplogroups. The results were baffling. Certain maternal lineages corresponded to regions thousands of miles away, across oceans, in areas with no known prehistoric contact. Conventional migration models, land bridges, coastal navigation, or trans-Pacific movement offered no explanation. The Paracas genomes seemed to appear where they should not have been. Even more puzzling was the age of the samples. Radiocarbon dating placed these individuals firmly in the first millennium BCE, a period when long-distance travel was assumed to be impossible for the scale suggested by the genetic findings. There were no archaeological signs of foreign populations, no evidence of intercontinental trade, no maritime tools capable of sustaining such voyages. Yet the genetic signatures were real, consistent, and repeated across multiple individuals, suggesting either an entirely undocumented migration or a mysterious ancestry that remained invisible in every other record. Researchers found that the Paracas elite were physically unique and genetically unusual. Their paternal lineage showed very little diversity, suggesting strict internal reproduction, while their maternal DNA included foreign origins. This points to a deliberately controlled lineage that stayed isolated yet selectively incorporated outside influences, defying usual environmental or cultural explanations. Haplogroup mapping revealed sequences common in Central Asia and even hints of markers associated with distant Eurasian populations. These were not minor deviations, but defining genetic traits, repeated consistently among the individuals studied. How these sequences arrived in the southern Peruvian desert without leaving any other trace challenges everything anthropologists have assumed about human settlement patterns. Traditional models, which map migration from Siberia to the Americas across the Bering Strait, cannot account for this anomaly. The data suggests a connection that bypassed the Americas entirely, leaving a population genetically isolated yet linked to distant regions. The implications for understanding human history are staggering. These findings suggest either an unknown transoceanic migration that left no archaeological trace or the survival of a previously undocumented human population, distinct yet capable of long-term continuity. The Paracas elite were biologically engineered or preserved in ways society and science have rarely recognized. Their genetics indicate careful management of ancestry, reflecting an awareness of identity and inheritance that goes far beyond social ritual. The mystery becomes more urgent when considering continuity. These markers appear repeatedly across individuals spanning centuries, implying long-term preservation of a genetic legacy. This was not random. It was a deliberate safeguarding of lineage, maintained through social, cultural, and perhaps even technological means lost to history. These people were not only elite in life, they were deliberately exceptional in origin. How can such a lineage exist without a migration path history recognizes? And what secrets might their ancestry hold about the true scope of human movement in the ancient world? Let us take you through the secrets. The evidence history cannot absorb. The Paracas elite had already upended expectations about social structure, physical form, and genetic ancestry, but the final revelations forced historians and scientists to confront an uncomfortable truth. Some evidence is too disruptive for conventional narratives. Once the full scope of mitochondrial and Y chromosome sequences was analyzed alongside cranial morphology and burial patterns, it became clear that these people did not fit into any established framework of human history. Their existence demanded a rewriting of assumptions about who migrated, when, and why. Institutions struggled to process the implications. Museums, universities, and archaeological bodies were confronted with data that contradicted centuries of accepted scholarship. Scholars who approached the findings with caution often face skepticism from colleagues unwilling to consider that history might have overlooked entire populations. Even the Paracas burial site itself seemed deliberately designed to resist easy interpretation. The tombs were sealed, the elite were isolated, and their culture left few contextual markers outside of textiles and ceremonial objects. Every discovery amplified the sense that the historical record had intentionally or accidentally omitted the full story of human variation and migration. The skulls, once thought to be anomalies of cultural cranial deformation, now revealed a population genetically engineered or preserved over centuries. Internal bone structure, sutural patterns, and cranial volume suggested traits that are rare or unseen in Homo sapiens. These were not one-off mutations. They were consistent across hundreds of individuals, spanning generations. Combined with genetic markers pointing to distant continents, the data indicated a level of biological stewardship unprecedented in pre-Columbian South America. It was not simply a question of ancestry, but of design. The Paracas elite represented a population deliberately maintained, both physically and genetically, in isolation from surrounding societies. Textile and artifact analysis reinforce this narrative. The precision, quality, and uniformity of fabrics, ceremonial items, and exotic materials suggested a culture that understood preservation and control on a societal level. Each burial was a message. Identity, lineage, and status were not ephemeral, but eternal. Even the desert's climate, wind patterns, and soil composition had been harnessed to maintain this archive. It was an extraordinary collaboration between humans and environment, with centuries of careful planning embedded in stone, fiber, and flesh. The broader consequences of these discoveries extend beyond anthropology into history itself. If populations such as the Paracas elite existed with such genetic and cultural distinctiveness, then accepted timelines of human migration, cultural exchange, and evolution require reassessment. How many other societies, innovations, or genetic lineages were overlooked because they did not leave visible traces in familiar locations? How many mysteries remain locked in other deserts, mountains, or buried tombs, their significance lost to conventional frameworks? The Paracas findings challenge the very notion of historical completeness. Moreover, the resistance to these findings underscores a larger issue. Human knowledge is constrained not only by evidence, but by expectation. When data conflicts with established paradigms, the reaction is often suppression or doubt, rather than immediate reconsideration. The Paracas skulls and DNA sequences did not fail because of methodological error. They failed to fit comfortably within the world modern scholarship had constructed. Their preservation, separation, and extraordinary genetics demanded that the past be reimagined in ways the present is only beginning to entertain. Even beyond technical and institutional considerations, the Paracas elite leave a profound moral and philosophical question. What does it truly mean to protect identity across millennia? For these people, burial was not a cessation, but a deliberate continuation of existence. Bodies, genes, and artifacts were meticulously curated to communicate lineage, uniqueness, and a rigid social order that outlasted immediate memory. The immense effort invested in such preservation signals extraordinary foresight, planning, and reverence for continuity rarely acknowledged in human history, challenging everything we think we know about ancient societies. In sum, the Paracas elite force a reckoning. Their skulls, their DNA, and the desert that housed them reveal a society operating on principles far beyond our understanding. They were not merely unusual, they were exceptional, carefully preserved, and deliberately distinct, leaving an enduring imprint on both biology and culture. History, as we know it, cannot fully absorb them, yet ignoring their existence risks underestimating the extraordinary breadth of human ingenuity, strategic foresight, and deliberate shaping of identity across generations. The evidence from Paracas does not merely expand our knowledge, it demands a completely new framework for considering humanity's past. For some mysteries, the real challenge is not in uncovering the truth, but in finding the courage to accept it and to re-evaluate the stories we have long taken for granted. Were you shocked by the genetic revelations hidden inside the ancient skulls and the questions they raise about human history? Share with us which discovery stunned you the most.